everyone. We're going to start. So this morning we have two sessions. First session is just an open discussion on topics that I ask you to propose. I have also some I would like to discuss. Then we're going to have a break. And after the break, we're going to split in a different room to have a discussion on the working group. So first important thing, you need to have to sign the attendance list again today. Don't forget to do it. One thing we forgot to talk about yesterday is the GDPR. So we took photos just to make sure that everyone agrees. We can use some of the photo on the website. So if you do not agree, just let know Yara about it. And let's start by discussing different topics. So one thing is the lobbying we can do on the European Commission to have a topic of interest in the next call. So I just would like to remind everyone how works the Horizon Europe call. So there is a strategic plan which is decided by the EU. The next one will be for 2025-2027. So the work plan for 2025 is already negotiated by the different countries last year. We should have a draft very soon. It's too late to add a topic. It's still time to make some slight change, like adding some keywords on this. That could be very critical because adding a keyword could allow to have, for example, some of us adding in some topics where we would not be if there are no topics. I remember 2 years ago the one was on root trait. It was a lot on root architecture, nothing on microorganism, and at the last moment, I don't know who has added the keywords need to be interaction with beneficial microorganism. That's open an avenue for us to be in this topic. So the draft should be sent by the European Commission to the ministry in the next days. Then the ministry will or not send it to institutes, universities, and finally to researchers. Often we have a very short time to read them and make recommendations. It could be just a few days or no days if it does not go fast enough from the ministry to the researcher. So one thing I propose is they might go at different speed in the different countries. So if any of you received a draft for 2025, you could just share it with the other, because it could be faster in one country versus another one. And then we can just discuss by email if we find any topics where we can propose to add some keywords that will be the call this year, this summer. Where we can make more lobbying will be on the work plan 26-27. Nothing is decided now. So they start to collect topics in the countries. For example, we received information from INRAE in France. They want the topic by end of May. They're going to discuss it then in the French ministry. And then the representative of the French ministry, together with all other countries, will go in Brussels to debate about topics for the working plan 2026-27. So there are in Horizon Europe there are 6 clusters. The cluster 6 is food, bioeconomy, natural resources, agriculture, and environment. That's the one in which we feel the best. And this work plan, they are organized by program committees. So there is one per cluster. And in this program committee, there are representatives of EU and of each state member of EU. So our way to do lobbying is through our own employers. That's them are going to discuss at the level of the ministry. In France, it's going to be the agriculture ministry that will be in charge of having a representative for the cluster 6. We can have also a direct, try to do direct lobbying to the program committees, but it's not easy. We don't know them correctly, except if any one of you know how to go directly to the program committee. But I think the best way is going through our institute. But what is important, I understood on the program committee, is if a topic comes from different country, the chance it's adopted is much higher than if it's only proposed by one country. So it would be great if we can, in each of our country, agree on a topic, send us to our representative, and then if the topic comes many times in the program committee, we have more chance. So do you have any idea of how does it work in your own country? Is anyone aware of this? Have you received some information?
In Albania, the agency that takes care about Horizon let us know about it.
They ask you also to send a topic? No.
They just in this moment let us know that the next program is open and about the topics that we cover at Cluster 6 to have a look.
Okay, but this is probably for draft 2025, because the 26-27 is not done yet at all.
What? No, it's 2025.
Yes, okay, so you have received the 2025 draft?
I'm checking right now, in fact,
yes.
The email. It is a call for Horizon 2024, Cluster 6, opportunity on founders and tenders.
Yeah, you can look, and if you find the 2025.
We can check together also.
So if you don't know what the process in your country to propose topic, I propose you ask to university or, yes?
Yeah, so just to clarify that it's not clear. So in France, for example, the INRA, they make all this, so they share with people to make some topics and to discuss them at different levels. But me, I belong to the CNRS, not the INRAE, and I have no information through my employer. So it really depends. You have to find a good way. So it can be the universities, the employers, some associations, some things that make some lobbying at the European level. It's not easy to find the way.
And actually in RIGHT, first year they informed us this way. Last year, you remember, we had to find who is the person in charge in INRAE for this. They were doing it, but without telling anyone. So we had to find who was doing it. And actually, if we don't do it, some other are doing it. And they will have their topic.
Yeah.
So it's very important. Yes.
So yeah, they do it in INRAE for 20 years or more years, and sometimes they send it to everybody, sometimes they don't send it. Sometimes they change the person in charge and you lose the contact. So.
Definitely the topics that are in the Horizon Europe call, they are not there just randomly. It's not the committee program which decide by itself. It's really researchers who have made lobbying before.
It looks like closer to Brussels you get the information already.
Yes.
Maybe Bart has something, so.
Well, I will forward you the email, so it has already the strategic plan.
The 2025 or?
Yeah, 25 to 27.
Okay. So we can then spread it and each have a look and.
So I received it already like 2 weeks ago.
Okay.
Please raise your hand before asking because of streaming.
Yes. So Stefan, I think it's.
Yeah, I can also talk like that if that's okay. So occasionally, because we have in my institute, I have to say it like that, strong people when it comes to EU business. And sometimes I think these pre-draft for the now active calls I saw also on my desk. And what I would do when I'm back home, I will approach some people at SALV, which is only one out of many institutions in Germany. And I don't know, if we have some text in this regard, some text element, I can then push some people and say, can you bring that to the National Contact Office, something like that. But yeah, there's no guarantee that it goes in. But we had that 3 years back, that suddenly you got a text, as you said, to all the main researchers in the institute, and we had 3 days to add something, and I was completely overwhelmed. What the hell should I add here? No? But if there,
So yeah, so exactly what I said. What you can look at if there are topics where it could really fit to have some beneficial interaction, but it's not specifically mentioned, then like root traits, what a perfect example, you can add it,
Yeah.
and that's useful.
And
Or like decrease, if a topic is decrease of fertilizer, I don't know, biocontrol, but there is nothing about beneficial soil microorganism, this could be added.
maybe last comment. So if you or someone would put some lines together, some keywords you would like to see that could help, couldn't it?
Yeah, I
Because
can
based on the
document like kind of topic in which we could add some keywords
Yeah.
if we see them. Yeah. Because there are some people online that need the microphone.
So through the cost officers or the national contact point, you think maybe it's a way to ask them what is the, in each country, what is the process to get access
I'm
to the
not sure they
topic?
know how it's organized in each country, in each institute.
Even the national
Maybe.
contact points or the cost?
For the
Maybe
cost
it's
of-
a way to ask this, huh? You never know.
For the cost officer, we can ask how we can reach directly the committee program eventually, but in each country, I don't believe that. I discussed this one with Lucia last year, how we can make lobbying. No. Okay, so please try to find how you can provide a topic to your own institute, and the second part is which topic can we send it? So you have probably seen my email. I wrote a draft of something which is written in the way where they appear in the call. So you always have this title, expected outcome, scope. So the expected outcome is something long term. It's not something, it's a general context in which a research is done. The scope is more the objective of the project itself, and usually it's written this way, proposal should, and it's here you can place something that need to be there and which allow us to make sure we can fit in the topics. The debate is how restricted should be this topic, whether it should be very large with a risk there will be a lot of competition, or whether it's there is high chance that it's selected by the committee program, or it's quite restricted. We have good chance if there is a call to be on it and to be funded, but there is a lower chance that it's selected by the program committee. So, for example, I wrote biocontrol, and I think Anna, you also comment on this. I had this discussion with the INRAE person who are in charge of the European policy, and yeah, they suggested to keep it large. But there are so many consortia that can do biocontrol that somehow it's not very specific to. But if I think I put enough of things really specific to the beneficial microorganism with as well nutrition, that we can also add the layer of biocontrol, or maybe it can be written another way, like say stimulating resistance to biotic stress, and not using the term biocontrol itself. So I encourage you to read this draft, make some change, comments, add keywords, or just tell me whether you agree with this, and if you agree with it, just keep it and send it to your university, institute, or directly ministry. Eventually, if it's in your country, you have to go directly to the ministry. And I think in France, at least, they told us that, for example, when we propose such kind of thing to INRAE, which is going to discuss with the ministry, if we claim that this was already discussed at the European level, for example, in the coast, they will be more willing to push it because they know there will be consortia beside. If it's only one person who proposes a topic on his own research, but it does not have a potential consortia to make an application when the call is there, they won't do it. They want to know that people are ready to go if the topic is written in a call. So it's important to claim that this is not made by only one country, but it's made collectively by a researcher network.
Maybe I'm not used to the procedure, but I just got an email 2 weeks ago regarding feedback opportunity, a survey from EU actually for the strategic plan 25. I just forwarded it to you,
Okay.
but it's in form of a survey feedback. I don't know if it helps in anything.
So regarding this outline, I think that we need to expand a bit the remit, including something beside the actual science that we may pursue, because very often these European proposals are broad, including, again, social scientists, economists, citizen science. So you better start in considering how to integrate, in light also of what we discussed yesterday and last night, because this reads a bit too sciencey for the type of proposal typical are put forward.
Yeah, it's a good point. Yeah, because definitely what was told by INRAE, it's those who decide at the end, they are not scientists at all. They are just politics. So it needs to be very clear how the impact is for the society. And so it's a good point. Yeah.
Hello. Why don't you propose a call instead of a topic?
What do you mean a call?
A topic is related to the call, right?
Yeah, in the call you have many topics.
Yes, so why don't you propose a call? And then you will have more opportunities. And the call will include more topics.
Mm.
Because your topic is now related to the call, right?
Yeah.
So it can only be proposed if the call is related to our topic.
There will be a call in the cluster 6 for sure.
Yes,
so in the next tomorrow,
but calls are usually, they don't always include this kind of topics.
So I think that it's in each work program, you have cluster 1, 2, 3, 4, 5, 6. So
yes.
there will be the 6 for agriculture. And there are going to be many different things inside of the 6. So I don't see what is the... I'm not aware of how we can propose a call, specific call.
Maybe. I mean, you didn't know you can propose a topic. So, I mean, you assumed you can propose a topic, but maybe it is possible. Because then we have more chance. And like this talk, the topic is rather narrow area. I mean, just a thought.
Yeah, I'm not aware that we can do something else than just proposing topic that goes in the call.
Okay. But now you don't know what the call will be, right?
But it's always the same. So it's each year you have the Horizon Europe call with each cluster.
Because in our topic, as you can see in this now in this meeting, there are so many directions that we can go. And if you only have one topic, then only 25 to 30 institutions can participate, right? And there are so many of us. So why not proposing a call, and then many of us will be able to participate?
When you have a topic, sometimes you can have several projects funded. You have at least 1 or 2 or 3, oh sorry, not 1, at least 2 or 3 projects that can be funded on one topic. And then there might be more applications, as always, and then there might be a competition included between some of us if there are several applications.
Okay. Another thing, since I'm having a microphone, that was on my mind, we didn't really discuss the legal issues here, the legal points of registering biopesticides, biopromoters. So maybe just a thought that we also need to include this, because in Europe regulations are rather strict, especially if you have to go through EFSA, and that is something I didn't see we mentioned here. Thank you.
Just to make a point, we are going to discuss that in the group number 4.
Yes. And
because that is where we are related.
there is also probably something we're going to discuss in the workshop that's going to be in September, which is about inoculants. So regulation goes with this, which will be more together with companies and other stakeholders, policymakers that we want to invite to the second meeting.
To finish on the topics, INRA, they ask us for topics, and I think the idea is to really make lobbying, and that the idea is that INRA is involved in as many as possible funded projects later on. So that's why it has to be specific, because you propose something that you will be able to be then in the consortium that will answer to this call. So if it's too general, they are not interested, because it's not the specificity of France, or even there, it's the specificity of INRA. So, and the CNRS doesn't do it, so it's really even under national scale. So it's lobbying, and some do it, some others don't do it. Some do it efficiently, others less. So that's why you have to find your way. Maybe also it's to ask colleagues that you know already participate regularly in European projects in your country. They may be more aware of how it's going. Maybe they will not share with you, but that happens really regularly.
Okay, let's discuss about other things now. So I received several points that could be discussed. This is from Luisa. This one, so it's many technical questions, I would say, about, you want to say about it?
You asked for some topics possible to be discussed together. So in my mind, with my experience, I would say, yes. So the first one was dealing with the sequencing approaches. We are all dealing with the description of biodiversity, of the plant microbiota, or the soil microbiota in different contexts. And so I was wondering whether time is ready to discuss and to open discussion on the sequences approaches that we have so far available. So moving with the idea to move from the simply taxonomic description of the diversity to the functional diversity. And so maybe we could see whether the metabarcoding is still a good approach, or we should move to the metagenomic approach, which is much more complex to analyze, as I had the chance to discuss already yesterday with Davide. And so this is, for me, is an open question. The second point was about the culturomics. So especially, I am from the arbuscular mycorrhizal fungi side. And so I know it is very difficult to make pure culture of these obligate biotrophs. But I think now it's time to maybe put an effort in trying to isolate more AM fungi from the field, because in available collection, we have just a few of them. So this with the idea to really explore the potential also in let's say, applicative perspectives. And beside this, beyond AM fungi, I think that maybe some methods could be developed or already in place to better characterize fungal and bacterial strains, especially looking for these plant growth promoting properties. So I think this could be one other point to be discussed. If I may go on, and then one more point I suggest is the to make maybe some thinking about the strategies to obtain more sequence genomes from beneficial root-associated microbes. And so not only EM fungi, because this is my field, but to extend maybe also the genetic resources we have on these beneficial microbes. And I would extend maybe talk about also additional genomes for fungi in particular. The recent experience on described the viruses living inside AM fungi, and also AM fungi possess and harbor endobacteria, so this could be considered as additional genomes and may provide additional traits to the fungal host that may have an impact also on the plant host. And that's it. This was just some thinking on possible topics to be discussed here together.
I also did a question that Tanya asked yesterday. It's, what would be the best source of AMF inoculum for experiment? And what's the stability also? She said she's producing the strain in vitro on her lab, but at a certain level she lost the strain, so how we can manage to keep the strain. And just to go on this, I had quite similar question as well. How we can coordinate, a bit specific on AMF, because also that's my topic. How we can coordinate effort for having more genome sequence, which strain to sequence, so we can discuss also if we have a collective effort with trying to sequence what and propagate. But sequencing means we need to amplify them, have pure DNA. Which technology to sequence? Probably we want long read sequencing, so we need high molecular weight DNA. That's also which protocol could work with AMF. How to organize transcriptomic to have genome annotation, also good quality, and how we can have funding for this, or can we work on our own budget. But when you start to add sequencing DNA plus RNA, there start to be a cost associated. Concerning the metabarcoding, also the question, as you mentioned, so in between the current metabarcoding and the metagenomic, there is switching from the short read to long read. We discussed it a bit with Maria yesterday. So who is trying now to develop the long read metabarcoding, so with which sequencing technology? PacBio is making a good quality sequencing, but still quite expensive, and you need a lot of sample. You can think about nanopore, but there's still a lot of errors. That's a debate we can have. For genome sequencing, it's not a problem, but for affiliation, that can create some bias. Open discussion. And the other point is which database we can use, because all databases, they are designed for ITS or 18S, so if a long read, we don't have database, so we should create them. It goes a bit with genome sequencing, maybe to or amplicon sequencing, at least on a pure strain to enrich database. So. And another question we had with Agnes also in the lab is how we can quantify the AMF in the soil. What's the best approach to do it? Measure biomass of the mycelium, 18S copy number, measure lipids, or try to quantify glomalin. There are many possibilities. What's the best? It's not clear for anyone. So there are many points to discuss. They are a bit technical and bit because that's our interest. AMF, and I was wondering what's the best way to discuss this. So first, who is interested in this specific topic and how we can discuss it? Maybe not here. It might take a couple of hours. So should we organize a subgroup on this and maybe have either on another meeting a specific workshop, I don't know, one afternoon, for example, on this, or there are even several questions. I think the metabarcoding is maybe not the same than something specific to sequencing AMF. So if you have some suggestion how we can work on this, we can do forum maybe when we can just ask our question and exchange, replying to it, debate online. We can make Zoom meeting also on specific topic with the people who are interested. One thing is we need to know who is interested in which of the topics. So maybe we can make a survey also with all these ideas now, knowing who is interested to discuss about which topic, and then organize some forum and video about it.
It would be definitely useful also to spread it.
What's the second for to the microphone?
If it comes to such a specific topic, then it's really good also to spread it to other people because, for example, in the AMF community, there are definitely many people who know about it and would be interested, even though they don't participate in this action.
Yeah, and then we can ask them to participate. If they want to participate in this discussion, that would make sense. No?
Is there some of these points that were proposed as deliverables or written in the document?
No. But I think we should benefit of this network to share also this technical. But I mean, what we wrote is we also want to have like standardization of methods in the fields. It's not a deliverable by itself. I think we discussed this. It's difficult to have a deliverable where we agree on a standard method. So that's why we remove it from the deliverables, but still it's a goal of such network.
Because just a question of organization and motivation, it could have been a motivation to have a deliverable to provide. And for example, to make a little viewpoint article or something like that to propose
there could be
one way of doing this kind of thing. There are regularly you can see this in some research.
I mean, sharing know-how.
And it's the opinion of a group of people or of a kind of lobbying also. And of course, nobody will agree, but at least this group of people have found this minimal agreement on the way to do the thing.
I mean, personally, I found enough exciting to share with colleagues about the methodology, not to need more motivation than just learning.
Yeah.
But so it's very specific to AMF. So if you have other topics you want to share in subgroup like this, I mean, please send ideas, and we can just organize a group of discussion on other technology or interaction.
I would go for some online call probably, that everybody can join if they want. Maybe we can focus one call on sequencing on IMF. Maybe not everybody will be interested on this. And I agree also that we can invite to this call very experts. For example, Nicola Corradi, who is doing a great job in this long reads sequencing, so they could have a chromosome level assembly of AMF. And we are already collaborating with him for sequencing other AMF, so we are developing actually protocols for having good quality, high molecular weight DNA for these purposes. So I think if we found maybe from this list a few more defined topics, then for each maybe we can propose a call, online call, to do it in the next month, and try to discuss the topics within this call. And then we will see whether we go from this discussion. So.
Okay, just as a comment, so I'm very interested in this, how can you quantify, characterize microbiomes in soils? And then we have meta-barcoding, and the colleague already said meta-transcriptomics, metagenomics, whatever. And then I also observed over the years, because I'm a more bacteria and archaea-orientated microbial ecologist and not so much familiar with fungi, but then I worked with people in fungi business. And so I'm always, in my mind, think, wouldn't it be great to have methods in this sequencing field that can be applied to any of these organisms, also including protists and whatever. So for example, I have colleagues that do metatranscriptomics of full communities. They go through all trophic layers who can go until the nematodes or something like that. And I'm saying that because there's also efforts also in Europe, how can we standardize the biodiversity monitoring in soils? No? So completely other topic, but finding the same technologies. No? There's this Lucas thing and a protocol where they have, based on meta barcoding, something. So I would be interested to join such an discussion, but with the idea that it goes beyond AMF.
Yeah, I think I also second that, because if we start to separate again the areas, then we are going to go to the same issue. And I think if we have a very standardized method that we can follow all, not just one area, that also can help with the regulation, because then we can suggest that these kind of methods are used to analyze the quality of the products, because sometimes if you have consortia, you only don't have like a microbial inoculant or AMF. You could have products that can have a combination, but someone needs to test that have all these. So if we manage to do these kind of methods together, we can also help to the regulation to have these methods and do the registration of the products quicker.
Okay, so let's spice it up, the discussion. So, I mean, what I'm going to say is my opinion. I'm not saying that this is the way it should be. So, I want to address the discussion here. I think that the concept of standard and standardization applies very well to engineering and technology, and not so well to biology. The reason being that biological system, let's say in a way, they may consider a series of exceptions. So certain plants and certain soil may provide you with a DNA amount or type of microorganisms that are different from the others. So, and the very same protocol may not apply to the, let's say the very same protocol developed for one system may not apply efficiently to another one. So I think that we need to be aware of that. So the dream of, and again, this may be controversial, and that's, I emphasize, is my opinion. I think that the dream of standardization will remain a dream. So what is important is having a reproducible pipeline that can be applied, hopefully, to a series of situations that are comparable. Ending up with one-size-fits-all type of solution, I think that doesn't really fit the system we are working with. And then I'll be happy to discuss a little longer. But.
Thank you, David. I think this is an excellent point, actually, and I had a heated debate on the similar topic with Luisa this morning when walking here. And maybe it's good to learn from other subjects where this kind of methodological tests have been ongoing for a long time, and by the way, have not finished. So in the microbial ecology, we shouldn't think that we actually can jump over and we don't need to test. So I fully agree that we should not dream of one-size-fits-all solution, but it's very good to test different methodologies to be aware what are the differences. So, for example, in plant community ecology, they have had all these discussions about how large should be a plot in order to be able to catch meaningful plant diversity estimate. Should it be 1 by 1 meter? Should it be 20 by 20 meters? Should it be circular? Should it be rectangular? And these are all meaningful questions, and we also need to ask them about how to measure fungal diversity in soil or in the leaf or in the stem or somewhere else. So we just need to do these maybe slightly boring questions, tests. My view.
Yes, they are all excellent points. I also agree that this is something that might actually stay a dream, but I think there are some solutions which hardly anybody applies. There are internal standards. There are synthetic DNA that might contain motifs for bacteria, archaeal, protistan, fungal, which should be added to every extraction of DNA or RNA. I'm not aware of many people doing this. I know there are some exceptions, but I mean there are no standard or there are no perfect ways of extract DNA from all soils. It doesn't exist. There are kits which work well, but then they remarkably fail for certain soil types. The same way, I mean, for the amplicons, we have, Maria will know probably better, I mean, we remarkably failed to produce primers for all fungi. It doesn't exist. We know that those which are widely used, they don't pick up the AMF efficiently. So, I mean, these are things that are here. We should think, we should discuss, but they're things which I think are able to move us forward, at least bridging the gap between the Protestant eukaryote or the prokaryotic eukaryotic worlds, which really develop separately. But I think, you know, having a synthetic DNA with motifs of bacterial and eukaryotic amplicons that could be easily distinguished for anything else is one way forward. I mean, that's probably a little contribution, but this will, to some extent, solve the long-standing issue of the fungal-bacterial ratio, for example. That's just enigmatic and remaining for decades.
One comment I have is, I agree we should not split. That was the idea, to be able to think all together, but there are some technical issues that are specific to one microorganism. If you want to detect rhizobia, you won't be able to use the same primer on a nitrogen-fixing rhizobia than the other bacteria. Otherwise, you just have the species, does not say whether it's a nitrogen-fixing bacteria. AMF is the same problem, the general primer. Detecting fungi that don't detect very well the AMF. So you have, I think we should keep together at the end. If we want to make a project and a proposal, we should be able to study all organisms, the nitrogen fixing, the AMF, all the other microorganisms in a project. But for technical discussion, to choose a solution that can then be used in a future proposal, I think there could be some specific discussion, and after we share the conclusion of our specific discussion. That's my idea. And I think the benefit to be all together, it's at the end to be able to have proposal on everything. And whether we should go outside of microorganism, I mean, it would be important also for proposal, I agree, to have the whole biodiversity of the soil, but yeah, the topic of the action is to microorganism rather than other organism. But it's true that it's a bit pity that soil diversity is always seen from one side, either microorganism or other. It's already an effort to try to have all microorganisms together.
So I think this idea of organizing webinars, dedicated webinars, we can put a title on them: root benefit webinars on this and this and this, and specialize them, and then this can be also an outcome for the cost action. We can record the presentations, we can invite people, we can participate, each one of us, to the ones that we feel that we are more interested in, and can be organized in a regular manner. So every 2 months we are going to have, for example, every 2 months we are going to have the next webinar on this topic, and we can identify more topics than the mycorrhiza, for example. We can discuss this in the recording.
No more comment on this part? Okay, so the next topic was proposed by Isabel. Isabel, can you comment on what you're saying?
Thank you. I just raised a question that I believe it's a question that many of us felt. That's the problem of the level of literacy of the stakeholders, including farmers, inoculum people that are on business, and so their lack of literacy or illiteracy regarding the use of beneficial microbes in agriculture. And so we do know what are the benefits, how those things work, but apparently we are not able to transfer that information in a way people can understand and use, and understand this, the use of these microbes can be useful. So what are our knowledge transfer gaps? Why are we not reaching the end users? It's a matter of the forums we use to speak. It's a matter of the kind of language we use that is not understandable for them. It's the not being able to show that, in fact, these microorganisms can be effective and useful in the production process. So what's in the frame of this group or others, what instruments do we have to address this issue? What can we do to overcome this problem? Should we have specific forums, training schools, workshops, whatever, to pass the message or try to pass better the message? Should we try to write in broad spectrum magazines in order to address a wider public? Should we try also to elucidate policymakers, so when they are making the rules and the laws, and to be themselves also more aware of what is involved in these processes and with these organisms? I don't know, this was just kind of launching the discussion. Of course, within Working Group 4, these are subjects that we are addressing and will be discussed this afternoon. So once again, I invite you to come to the discussion on this working group, but of course, hearing the opinion of everybody, and it's important.
Writing a publication for general public, I think this is something we put in the proposal as well. We will have to do it at some stage. Whether it's enough to reach end user, I'm not sure.
So wider public for sure. Reaches and a wider public.
Yes.
I'm just going to speak for my experience, and what has helped me is to go out of my comfort zone. So I was very good like at Rhizobia, my own circle and everything, but actually I start to approach alliances, and I'm talking, for example, in Denmark, and also because I didn't know the ecosystem of Denmark. I'm not Danish, so I needed to learn this. So I approached the Alliance for Biosolutions, because they have been working already with several companies, but also with the policymakers. And first I asked them, what do you have there already? What do you understand about a product that needs to be there? How is the process of the regulation? First I asked them, it's like, okay, what do you have there? And what do you need from us, the scientists, to give you to actually transmit that better with the policymakers? Because I can't speak with the policymakers in Denmark. First of all, there is a big language barrier. Although they speak very good English, it's still better that they speak in their own language regarding the legal language. So for me, it's better that I go to them, ask them, what do you need? They ask me, and then I go with my comfort zone, like the scientists, and say, okay, we need to kind of help them with this part. So that is what has been helping me, at least in Denmark, and is go out of the comfort zone, but with a useful bridge. It's going to be impossible for us to do all that exercise, but I'm pretty sure in each of our countries, there is someone working already in that. So just try to approach to them.
That's probably also something we expect from the meeting in Portugal, to have some of the stakeholders that could tell us what they expect from us, which kind of result we can provide them that will help to use bioinoculant to be more careful about the diversity in the soil. But somehow, for me, it's very interesting, but it's not directly the end user. It's not the farmer.
No, they also contact the farmers.
Yes,
they
but
are
then we don't have the direct contact with the farmer. There is a layer in between, which can bias a bit what the farmer wants and what
these are the
breeders or the sell-off inoculants wants also. You know, there could be, but it's very good.
I just wanted to say from my experience from Serbia that producers have more success in spreading the knowledge to the farmers and the end users than scientists. So maybe there are better connection to farmers. And where I see a huge gap is between what scientists do and the research and development that is needed to get the product. So here I see a huge gap because for us, we need to publish in high impact journals, and there is a trend to work on fundamental fields, which when we finish our research project, it is still far away from what is needed for research and development for producers. So here I see a huge gap, and not as huge as between producers and end users.
I guess in different countries there are also agricultural advisors who are not producer or breeder, who have also direct contact with the farmers and can provide advices on the practices to use. But there are also, our experience, we try to go through them, to contact farmers, to sample in the farmers, and there are very different advisors. Some actually push to use some practices like no-tillage, cover crops, and some other are very classical advisors. So we also have to find the one which fits with the enriching the biodiversity and the use of ecosystem services from the soil. They are not all aware of this.
Just thinking about this topic, and from reading the Horizon Europe calls, obviously the focus on living labs is trying to address this and trying to encourage connection between the fundamental research and end users. So I'm wondering if anyone in this community is already involved with any of those living labs, and what that experience has been like.
I try to make a long story short. In my institution, we tried to get a new department with 100 people that is doing living labs by institutional funding. And the decision on that will be made this year. I personally only involved in developing these ideas. And there's these agroecology labs, and I know that there is at least one coordination. So one proposal will be submitted this year where someone of us is coordinating. I don't know if that helps for us too much, because the term living lab means very often more small technological innovations, co-innovations related thing, like a chemist has an idea that is relevant for product development, and then a company comes and that is. So we think a living lab is much larger, it's much more complex, it includes any stakeholder from the politician to the local farmers to NGOs and whatever, and it also includes social innovations in a region. And I think there was in INRAE some work on scenarios for pesticide-free agriculture, and there was also one scenario proposed by others in Europe that goes also in this direction. So I don't know how that relates easily to what we are doing here, but my feeling is there's a lot of research money on European level now going in this direction. So my comment on that.
Yeah, I just wanted to comment on the same line as the last speakers, that I believe, well, we have a problem on short projects. There's not so much long-term research. We get funding for a few years, and then how to reassure the stakeholders is sometimes to provide long-term data. And I think that's a gap. I don't know if it's us, I don't know if it's the Living Labs, I don't know if it's when you want to bring a product to the market, but we need to be able to reassure somehow that maybe 1 year would not be amazing, but we'll have less variability, or that, and I guess until we can provide that they will want to hear this kind of data in long term, but then we need long-term funding for this.
About the Living Lab, you have a comment?
Thank you. I fully agree with you, because the projects are financed in 3, 4 years, 5 years, and we don't have the continuity of financement sometimes to continue what the projects that should produce reliable data. Regarding the Living Labs, in my university we are involved in one Living Lab with stakeholders, research institutions, and the point is this might be a way of spreading more, have new forums to spread information, but the point is that in the point of view of the stakeholders, people who are involved in these Living Labs are already people that are aware of things and want to participate and learn more. So it's not the kind of, it's the public that we have already touched. So we have to go in a broader spectrum, I would say.
There is an ongoing call about the Living Lab, no, by EU to reinforce yeah. Okay, let's go to next. Next topic was proposed by Joel about our carbon print.
Yeah, I'm feeling a bit guilty each time I'm traveling with an airplane, and also my institution, and I guess most of the institutions in Europe are setting up plans to reduce CO2 emissions. If I have more than 12 hours travel by train, I'm allowed to take an airplane. And we have also now quotas per institute, which means that we need to apply about 4 months in advance for a flight. And it could be that it's actually rejected if you don't do a presentation, a poster, if it's not international. So you have to have good reasons to travel with airplane to justify it. We had already this in previous cost actions that I was involved in. Cost didn't bring really some solutions to that. I cannot deny that there's an added value with networking, but we produce also a lot of CO2 with this. And it's also nice to meet each other face-to-face and not online. We proposed some options in a previous cost action where we would have small hubs of countries. Let's say the Baltic countries would meet each other in Riga, for example. They can, maybe not the best, but they should be able to travel with train. The same maybe with Switzerland, France. We would meet in Paris maybe or something like this. And then we would meet online with other countries to minimize as much as possible the CO2 emission. It doesn't bring the same as we have here meeting with everyone, but it's one solution. And we've been through two cost action in my case during COVID, and it worked, but I must say the network was established before, so we had already face-to-face meeting, and the last 2 years were just online, and it worked. Of course, it was not the same as having face-to-face, having a beer, sharing a beer or something together like we had yesterday at dinner. But yeah, there might be solutions. I was really happy to see that you went for a training school online in June, so that is really a good solution, I would say. But if there are any other ideas, I think it's really important. Brussels won't really bring something in this direction. It's probably a drop in the ocean, but every drop counts, I think.
It's really a pleasure to see this topic on the screen, but we are missing the actually really important things. A few days ago, we were trying as an entity, private entity, to try to find CO2 footprint for microbiological fertilizer and to try to measure the both sides, so the production and the impacts on the fields. So, at least at our case, answer was that there's no model to measure impact of the microbiological fertilizer and capturing CO2 in a soil, degradation of the organic matter and so on, and try to measure that, or at least to implement the model. So the answer was there's no model and it's very expensive and so on. Maybe some of us know the model, or it's on the way to have the model, because it's crucial thing if we speak about microbes and soil and CO2.
Yeah, we shall include this in the working group 3, exactly this. So we try to establish the models, but first of all we have to get the numbers, and they are not easy to get.
Probably you are aware of some research project on this in different countries. I know that I was involved in one which was not funded. I know that other people in France, at least for France part, are trying to do it now. Carbon sequestration through AMF at least. Including how colonized roots versus non-colonized roots can be degraded. There is the biomass itself and the degradation of the organic matter from the roots, colonized or not. But yeah, we are far from having the numbers yet. So what's opinion? Yeah, I fully agree. Also, I was a bit ashamed at the beginning. We try each on our side, I guess, to reduce our carbon print, and we have all these meetings now, but we cannot avoid at least some face-to-face meeting for the first year. I think even the second year, we can think about it for the third or fourth years, whether we can have an online meeting at this stage, but we know to each other well at the beginning. Yes.
Has anyone calculated that option, bullet point number 3 is really offsetting more carbon dioxide than emitting in one place? So there was a calculation behind that? Or
it's just some ideas. It's just some idea we proposed 6 years ago in another cost action, how to reduce. And we came to this idea that we would make in small hubs. Maybe there's better location than others to organize meetings.
Yeah, that's the point.
I'm not a meta-analysis guy. I'm going to follow this course.
Training school, so maybe I can come with some options later on, but honestly, I have no idea.
Definitely our choice of location of meeting could have an impact, whether it's really in the center or in the side.
Yeah, I agree also with this idea. We have also the 12 hours principle in our institute. I think it makes sense to have some kind of a borderline, and what we saw yesterday were the points in different parts of the cost action. So as soon as all the people have been more or less activated themselves, or other people have activated them, we will probably find these kind of hubs where we could have also some meetings. So partly online, partly in presence, but of course moving around Europe, yeah.
If somebody said it already, but also ways to group with other different events together. So if there is a congress on beneficial microbes or on mycorrhiza, So I think even in the community and the Rhizobium and mycorrhiza, there will be effort to coordinate this meeting. So then you stay longer, but you have more activity.
Due to the distribution on the map that David showed yesterday, maybe it should be calculated a point for the next year meeting where to organize the and to calculate the lowest place to reduce CO2 emission. And it
we
could
also
be
have to find
useful.
a local organizer who are ready. Because if we
Yeah,
decide
also saying this, maybe I'm a local organizer that could contribute through organizing in Albania. But should be calculated if Albania is the best place for reduction of CO2 emission. And another...
Is anyone aware of a system to calculate this? I'm sure there are some websites.
You are not the guy of meta-analysis, but maybe you can help.
Is there a website where you can check your CO2 emission for a flight, and maybe this?
If you could
Yeah, I have noticed that it can be done, but we have to check all the members from where they come and to make a calculation. And another thing, it could be also mentioning the topic of the horizon that to use bacteria or microbes to reduce CO2 emission. So their effect on climate change. If it has a connection on it, I'm just dropping the
that
topic.
was the point just discussed before, the carbon sequestration by microorganisms, yes.
When we're talking about CO2 emission, I think we should also then always include N2O. So greenhouse gas emission, let's say.
Travel, yes, yes, it should be G.
Thank you. This might be a bit controversial, but in terms of the CO2 emission, we should also consider how much sequencing we're doing, because generating all the data that it needs to be on big servers all the time, constantly available, and if it's just a poorly set up experiment, we also contribute to this. So I think these plane tickets once a year is probably a little bit, it's important, but I still think there are also other considerations we could do and planning our experiments carefully.
Field experiment versus glass house or growth chamber also make a huge difference in terms of CO2 emission. And we also, that's just another point, we were told, especially the last years by in RAE, the cost of energy was increasing, and the message was start to think about the kind of research you will do in the next year. You won't be able to keep using growth chamber or greenhouses that consume a lot. In the next year, it will become too expensive. So, just to comment. Okay, another point raised by Joel the Nagoya is the Nagoya Protocol.
Yeah, it's reaching a bit what I heard already this morning regarding the registration of products, but also keep in mind that if you exchange resources in the cost section, make sure at least that you have an MTA and you keep track on what you send to who, because your country might have signed or ratified the Nagoya Protocol, and that is very important if it comes to fair and equitable sharing of the benefits. If I'm correct, normally the sequence of the genetic resources are also included in this. This is a bit the gray zone. I would say in our case, we always put it in the INSDC database, so NR GenBank. But there is some stuff in the background, and it's becoming more and more of a topic, especially in my institution. We might not even be able to perform the work if the Nagoya Protocol is not actually covered in the MTA or in the agreements when we receive strains. And I think here in this cross-section there might be a lot of strains, bacteria, fungi that could be exchanged in STSMs or in other work. So keep this in mind when you exchange the strain, to keep track exactly to what you send to. Also, if you plan to do sequencing.
But I think the big problem here is that nobody can give clear answers to this Nagoya Protocol. Because they will say if the strains are from before that age, then it doesn't apply. And we even have a specialist on Nagoya protocols at the Institute, but still he cannot give really clear answers, black or white. No, it's always something like, so it's confusing.
At least is the Nagoya Protocol clear for everybody? Or it's unclear for everyone?
If it's not clear, at the bottom of the slide you have actually the link that you can check. It's a bit a long one, but I think you can shorten it, and then you will get information when your country ratified or not the Nagoya Protocol. And you should have also your local national contact point for more details. I'm not sure they can really inform you much more about that, but I would say this is coming, yeah, this is going to be more and more hot topic in a way.
Yes, and we can be at least try to be aware of what our institute are requiring these days, just to be at least having no problem with our own institute when we share material or receive material.
May I just add something? Just to add something that some countries ratified Nagoya Protocol, but the rules are not included in the legislative. So we have a similar case. For example, Serbia ratified Nagoya Protocol, but it is not included in our legislative. So that means that we do not have the rules, and it's like it is not ratified.
Another point we started to discuss yesterday is which kind of skill are missing in the network, and which kind of effort we can try to do to attract some people, at least in some meetings, if we want to discuss this. So we already discussed a bit soil science, geneticists, economists. Then yesterday evening, and also right in the point, or related to the point you raised, Davide, sociologists would be important, at least to have some contact for in proposal. It might be important. And in terms of organism, I also think in between, something we have not heard a lot about is Mycorrhizae, fine root endophyte. They might play a quite important role in the ecosystem, and they are completely overlooked in the metabarcode analysis, for example. They're just not detected with the primer. I think it's a problem of PS, of copy numbers. They are very rarely detected, no? There are some actually good team in UK on this that could also fit in the network. I don't know in other countries whether you identify teams working on fine root endophytes. Ah, yes. Yeah, you see them. So they are abundant, and surprisingly, but they are not detected. So it's definitely a methodological problem.
About the first topic, so I'm involved in an interdisciplinary program about agricultural transformation, and there we are working with economists and sociologists. So I think the area where we can integrate with them so that there are no knowledge gaps are actually the ecosystem services. So we should not think that our mechanistic details or whatever is of any importance for that kind of calculation. So it's just it's a way to restrict the discussion that you. But at that level, we have found some common terms in discussion, and I found it fruitful, but rather slowly.
For the working group 3, definitely that's where they would fit. And in proposal, if we have proposal also.
Also for the soil science, the second member of MC, Maria Pazuchadol, she's originally soil scientist and works at Institute for Soil Science in University of Ljubljana. So I think, this for sure is expertise in this, which is in the action already.
Hi, I'm from Macedonia, and my institute is not very favorite culture. We are institute for tobacco, so we work from the seed, forming the seedlings, until the end. We produce seeds for tobacco. So in our institute, my lab is soil science lab. We have economics, genetics, everything that is related to tobacco. We also do field trials where we test all these products, but only mostly commercial products. So we can help with those missing skills. But we only work with tobacco for now. We have resources to do other, because we have a green field, greenhouse, sorry. So I think that we can do this.
I think the economists, sociologists, or researchers in soil science, their expertise can be used for any crop. If they are willing to do it, there is no specialty. The question is the willingness of the people to be involved in the project. Okay, so we are almost at the end of this session. Yes, the last slide was also proposed by Joel, just to introduce the other action, which is quite close to ours, in which Joel and Anna are both MC member of this one as well. So maybe you wanted just to advertise your poster also. Maybe some of you have seen the poster of the action.
It's just dissemination activity, as you know now from COST. So indeed, I brought a poster. It was a bit last-minute poster made by the chair, but I was happy to bring something here. We have a website, of course, like here. We started exactly on the same year, so October. We have exactly the same date. There's a huge overlap with this COST action. We had to rename our COST action acronym, let's say, to microp biomes, because the name crop biome was already taken. So for the name we changed it. We are present on X, formerly Twitter, if I'm correct, LinkedIn, and maybe other channels. So social media are very important, but we are a bit lagging, I would say, compared to this action at the moment. So things are being set up. We have the first training school on dysbiosis and microbiome functionality changes under biotic and abiotic stress. So it might be relevant for some people. And we have the first annual conference planned quite late in the reporting grant period. It's actually going to be in September in Portugal. And I think we have even since yesterday a third person in the room, Anna Maria from Finland, also joined as MC member. There's nobody from Latvia, if I remember well, so just keep an eye on eCost. You should have an account if you are here. Keep an eye on the COST action, see who is MC member, and might be still a place available for your country.
Okay, so after the breaks, we are going to split in different session room for different working group session. So one important point is to know how we're going to spread. Yes. Last slide was.
For the break.
Two and all.
Okay, you can. Let's follow the.
1, 2, 3, 4.
Yes, 1, 2, 3, 4.
Let's say 2, 2, 2.
Yeah.
Let's say 2, 2, 2.
Yeah, it's 2, 3, 4. Yeah.
1, 2, 3, 4.
You can imagine.
It's a mistake, but I cannot change. I don't know why I cannot write it here. One important point is to try to, that most of you have applied for different working groups. So now we would like to know who is planning to go in which session. So you can raise your hand up to have an idea of how many person wants to go in working group 1. Okay, working group 2. Working group 3. Look, then you're not going to be alone.
Yeah.
And working group 4. Great. So I think it's good. It's, so perfect. So we can have the break. I will leave the room number here on the screen, or can we go in the room from 10, 11? 11? At 12:30. There are classes. Yes. Yes. Yeah, we can.
Just for information, our conference will begin at 5 p.m. So we could suggest to go. Yeah. To go to the seaside.
The seaside?